Hi, hello students, good evening everyone. I hope all are doing well. Anyway, a warm welcome all of you to the Infinity Learn JEE YouTube channel by Sri Chaitanya. This is me Surya, your chemistry faculty from Infinity Learn. Okay, I believe I am perfectly audible and visible to each and everyone present over here. If anyone is watching in the session, please give me the hi in the comment box. Hi Atta, good evening. Welcome to the session. Okay, fine. We will be moving to the next part of our same chapter, biomolecules. Here we are discussing about the another type of biomolecules, that is about the nucleic acid, right? So before starting the session, I have a question for all of you. If you ever notice that some of your features like skin color, hair structure, something is resembles to your parents, right? Is anyone noticed that some of you have the same height of your parents or same hair structure, right? Hair structure or hair texture and skin color, right? What is the reason behind that? Yes, genes, right? Yeah, correct. Genes or genetics, right? So we will be dealing with some basic part of the biology along with this chemistry here. So in biology, you have studied about the cells, right? Cells. In the case of a cells, there is a small particle known as nucleus, right? Nucleus. In between the nucleus, there will be another particle known as chromosomes, right? We have already known about that. I think so. About the chromosomes. These chromosomes are the actual particles which helps in the transcription, right? Transcription means characters are transmitted from parents to offsprings or the children, right? Clear? So in the case of chromosomes, two biomolecules are present, proteins along with the nucleic acids. Proteins we have already studied, right? So in this particular session, we are going to discuss about detail about the nucleic acids. Can we start now? So let us start with the nucleic acid here. The nucleic acids, as you can see over here, chromosomes, right? Chromosomes made up of proteins and nucleic acids. The nucleic acids are the particles in the nucleus of cell, I have already mentioned, which are responsible for the heredity. Heredity means some of the characters or the textures, right, that are transcribed from parents to children. Right, so we are discussing about the nucleic acid here. When you are discussing about the nucleic acid, we can say that nucleotides are the building blocks or the basics of the nucleic acids, right? Nucleotides. So we can call nucleic acids as the polynucleotides. Clear? Okay, Adar, fine, it's okay. Anyway, all the best for your exams. Do your level best. Okay, fine. Okay. When you are discussing about the nucleic acids, there are two types of nucleic acids, right? One is the ribonucleic acid. It is known as RNA, and the second one, it is deoxyribonucleic acids, that is DNA. This is our genetic material, right? Genetic material in your body, that is known as the DNA. So, first of all, we have to learn about what you mean by nucleotides, right? What you mean by nucleotides? What are the major components present in the case of nucleotides? So, you can see over here, there is a heterocyclic compound, right? There is a heterocyclic compound present known as nitrogenous bases. Nitrogenous bases in the case of a nucleotide. Then the second part, it is a sugar molecule or else it is a carbohydrate molecule, right? A sugar molecule or carbohydrate. We have already discussed in our first class, right, about the sugar. The last one, it is the phosphoric acid or a phosphate group. These are the three components present in the case of a nucleotide. So in detail, we have discussed about one by one. We can start with the sugar molecule, right? Before that, there is a brief description about the three parts in the nucleotides. First one, it is the nitrogenous bases. These nitrogenous bases can be classified into two groups. What are they? First one, it is purines, right? Purines, and second one, it is pyrimidines. So in total, in the case of nucleotide or the nucleic acids, there are 5 nitrogen bases are present. As you can see here, the first one, it is adenine, right? Adenine and guanine. In the case of pyrimidines, there are thymine, cytosine and uracil. These are the 5 different types of nitrogenous bases. We will be discussing in detail along with their structure. Now, in case of pentose sugar, right? Pentose means it contains 5 carbon atoms, so it is a pentose sugar. In the case of RNA, it will be the ribose sugar. In the case of DNA, it will be the deoxyribose sugar. Then, what do you mean by phosphoric acid? What will be the structure of phosphoric acid? You must have an idea about the phosphoric acid, right? You might be studying in your P block elements. There will be a phosphorus having double bond O, then 3 OH group, right? So H3PO4E is known as the phosphoric acid. These are the main components of a nucleic acid or the nucleotides. Let us see sugar in detail. Here, I have already mentioned that there are 2 types of sugars, right? One is ribose in the case of RNA, another one it is deoxyribose in the case of DNA. So, when you are discussing about the structure of sugars, right? Structure of sugars, you can say that they are aldopentose sugars. What do you mean by aldopentose sugars? Aldopentose sugars. What is the main idea you are getting from the name of that particular sugar? Aldopentose. Aldo stands for aldehyde, right? Which means that this particular sugar contains aldehyde group as a functional group here. Then pentose stands for, or pen stands for, there will be 5 carbon sugar, right? 5 carbon sugar. So, and it is also present in the furanose form. While we are discussing about the fructose or the carbohydrates, we have already discussed about the structure of furan, right? The pentose sugar having an oxygen atom, that is known as the furan structure. So, when you are discussing about the structure here, you can see that there is a structure of D-ribose and deoxyribose, right? Is there any difference that are you observing here in the case of ribose and deoxyribose? Okay, just look into the first carbon, right? Not first carbon, just look into the second carbon, right? What you are observing here, in the case of ribose molecule, there is an OH group, right? There is an OH group, but in the case of deoxyribose, there is no any oxygen. There will be 2 hydrogen atoms above and below the plane, right? So, this is the main difference between the ribose sugar and the deoxyribose sugar, that deoxyribose do not have oxygen atom on carbon at the second position. On carbon at the second position, right? So, these are the main difference between beta D ribose and the beta D2 deoxyribose here. Clear? Then you must have idea about the alpha beta forms, right? While we are discussing about the carbohydrates, we have discussed about the anomers here. So, here it is the beta form. Is the thing clear? Fine. Now let us talk about the nitrogenous bases, right? The second part is the second term in the case of nucleic acid or nucleotide is the nitrogenous bases. From the name itself, it is suggests that it must contain nitrogen group, right? It must contain nitrogen. So that name comes for nitrogenous bases. Nitrogenous bases. Then we have already seen that these nitrogen bases or nitrogenous bases are divided into 2 groups. One is purines and other one is pyrimidine, right? Pyrimidine and purine. So we have already seen that there are five types of nitrogenous bases are present. These 5 types of nitrogenous bases are classified into 2 groups. So first one, it is the pyrimidines, in which contain cytosine. This is one of the nitrogenous bases. Then second one, uracil, and the last one, it is thymine, right? So you need to remember these 3 names along with their groups. It will be very important in your examination point of view. Which of the nitrogenous bases belongs to the pyrimidine or purine group, right? So these 3 are belongs to the pyrimidine group. Then the second one, there is the purines group, right? Adenine and guanine. Adenine and guanine are the 2 nitrogenous bases or nitrogen bases which is present in the purine group. Is the thing clear? So in terms of nitrogenous bases, we can define as DNA and RNA, right? In the case of RNA and DNA, all the amino acids, all the nitrogenous bases, right? Cytosine, adenine and guanine, right? Cytosine, adenine and guanine are occurring both RNA and DNA. When you are discussing about the structure of RNA DNA, you can say that there will be one difference in that. There will be the difference in one of the nitrogenous bases, that is thymine and uracil, right? Uracil occurs only in RNA. Uracil occurs only in RNA. Instead of uracil, in DNA, there will be the presence of thymine nitrogenous bases. Clear? If you are discussing about the structure of RNA and DNA, you can say that there will be 4 nitrogenous bases are present, but in the case of DNA, thymine is present. Instead of thymine, there will be uracil present in the case of RNA. Is that clear? So in terms of group wise, we can say that purines, right? Purines means they contain adenine and guanine. So we can say that in RNA and DNA, both the nucleic acids it contain or purines are containing as a nitrogenous basis. Clear? Hi Rana, good evening. Is that clear now? What are the different nitrogenous bases present in the case of nucleotides? Now let us see the structure or the images of the nitrogenous bases here. So here is the first group, that is the purine. As we already know, adenine and guanine belongs to the purine group, right? This is the general structure or general structure for the purine group. As you can see over here, nitrogen is present here, therefore it is known as the nitrogenous bases. So in terms of 2 different nitrogenous bases, first one it is adenine, right? First one it is adenine. You must have an idea about the structure of each of the nitrogen bases, because in the last attempt, right, the first attempt of JEE Main in January attempt, the question came from this topic, which means that you have to identify the structure of the nitrogenous bases. There will be the match the following question. There was a match the following question. So you need to pay complete attention to the structure of the nitrogenous bases, right? So as you can see over here, in the case of adenine and guanine, you can see an NH2 group, right? Amine group is present here. So some of the question may ask from which of the following nitrogenous bases that does not contain NH2 group. Let us see about the pyrimidines also. So this is the general structure of pyrimidine group. As you can see over here, this is a cytosine and this is thymine, and the last one it is uracil, right? So you can say that again in cytosine also there is an NH2 group present. So 2 nitrogenous bases that does not contain NH2 group are thymine and uracil. Clear? Thymine and uracil. And also while studying about the structure of nitrogenous bases, you must have an idea about all the functional groups along that the cycle chains, right? Whether it is belong to purine or pyrimidine. In your examination point of view, some of them may ask like that, which of the following groups contain oxo groups or keto groups also, right? Clear? Is that clear? So here is the base type along with their name and their systematic name, right? Systematic name. The first one, these three are belongs to the pyrimidine, right? First one, it is cytosine and its symbol is given as capital C, first letter of the cytosine. Then structure is already given here and the systematic name is here 4-amino, fourth position means there is an amino group, right? There is an amino group present in the fourth position. So, in the second position, there is a keto group or otherwise double bond O. So, it is known as the oxo. Then it belongs to the pyrimidine group. So, it is known as 4-amino-2-oxopyrimidine. Then the second one is thymine, abbreviated or the symbol is given as capital T, first letter of thymine. Then this is a structure. In the fifth position, there is a methyl group. So, it is 5-methyl. In 2 and 4 position, right? In second and fourth position, there will be the keto group or the oxo group. So, it is named as 2,4-dioxopyrimidine, belongs to the pyrimidine group. Then what about the uracil here? Uracil, the first letter of uracil stands for the symbol here, capital U. So you can see here, in the second and fourth position, right? Again, 2 keto groups or oxo groups are present. So it is known as 2,4-dioxopyrimidine. 2,4-dioxopyrimidine. Then we will be moving to the purine group. One is adenine, first letter A stands for the symbol here. Then what you are observing here, this is the structure of purine in which sixth position, right? This is the sixth position having an amine group or NH2 group. So it is known as 6-aminopurine. 6-aminopurine. Then what about the last one? Guanine, right? This is a nitrogenous base present in the purine group. So first letter, right? Capital G will be the symbol for the guanine nitrogenous bases. Then what will be the systematic name for this nitrogen base? Already number is given here. In the second position, right? Second position, there is an amine group. So it is 2-amino. In the sixth position, there is an oxo group, right? Double bond O or oxo groups. So it is known as, this is a common structure of purine. So it is known as 2-amino-6-oxopurine. Is that clear now? How we are naming the nitrogenous bases. Hi Shankar, good evening. Okay, is the thing clear now? So that I can move to the next topic. Just let me know in the comments. So far we have discussed about the sugar, right? Sugar which is present in the nucleotides. Then we discussed about the nitrogen bases. Then what else remaining here? What is the third part of the nucleotide remaining here? Phosphoric acid, right? Phosphoric acid, we have already seen that. Okay, phosphoric acid, we have already seen that there will be the H3PO4, right? This contains 3 OH group and 1 oxo group or double bond O. So it is known as the phosphoric acid here. So before going to discuss about the nucleotides, there is a one more term about the nucleosides, which means that in the case of a nucleic acids, right? In case of nucleic acid, if you are containing only nitrogen base, right? If you are containing nitrogen base and sugar only, then those term is termed as nucleoside. Nucleoside. If it is contained nitrogenous base and sugar only, then it is termed as nucleoside. Clear? Nucleoside. So, this is the structure of a particular nucleoside. You can see that this is our pentose sugar, right? This is our pentose sugar. In the first position or the 1' position, right? Okay, this is the 1' position or the first position. There is a base, nitrogenous base is attaching to the first position of the sugar, right? So, this particular bond is known as N-glycosidic linkage. N-glycosidic linkage. We have already seen about the glycosidic linkage, right? If you are discussing about the carbohydrates, there will be the glycosidic linkage which is formed by the release of water molecule, right? Similarly, here also, it is the glycosidic linkage formed between the nitrogen of the nitrogenous bases. Nitrogen of the nitrogenous base to the carbon in the sugar, right? So, it is termed as N-glycosidic linkage. N-glycosidic linkage. Clear? So, based on the name of nitrogenous bases, right? Based on the name of nitrogenous bases, we can name the nucleosides as adenosine, which means that the nitrogenous base contained in the adenosine is adenine, right? Adenine. Then, in guanosine, the nitrogenous base will be guanine. Then for cytidine, which means cytosine is the nitrogenous base here. Thymidine, thymine. Then uridine, which means uracil is the nitrogenous base here. Based on the nitrogenous base present in the nucleoside, it can be named as the following. Is that clear now? Okay, we have already seen that there will be the glycosidic linkage, right? There will be the glycosidic linkage between the nitrogen of nitrogen base to the carbon of the sugar, right? Carbon of the sugar. So, we may have some doubt whether in which position of the nitrogen, right? In which position of the nitrogen in the case of purines or pyrimidines, right? There are two types of nitrogen bases are present. One is purine and another one is pyrimidine. So, we may have the doubt whether which position of the nitrogen will bind with the carbon, right? So, if you are considering purine, right? If you are considering purine, what are the purine nitrogenous bases? Adenine and guanine, right? Adenine and guanine are the purine nitrogenous bases. So, we can say that the nitrogen at the 9th position, right? Nitrogen at the 9th position of purine. 9th position of purine is bonded to the first carbon of sugar by N-glycosidic linkage. Then, if you are taking the pyrimidine bases, right, like uracil, thymine or guanine, right? No, cytosine. Thymine, uracil or cytosine. If you are taking any of the pyrimidine nitrogenous bases, we can say that the nitrogen at the first position of a pyrimidine is linked to the first carbon atom of the sugar moiety. Clear? So, here it is the adenine, right? Adenine belongs to the purine base. So, in the 9th position, right? 9th position of this particular nitrogenous bases will combine with the first position of carbon in the adenosine. Then uridine belongs to the pyrimidine group. So, first position of the first nitrogen in the uracil group binds to the first carbon of the sugar in the case of uridine. Is that clear now? Now let us talk about the basics of nucleic acids, that is the nucleotides, right? We have already seen that polynucleotides. Nucleic acids are known as polynucleotides, which means that the nucleotides are combined together to form the nucleic acids, right? So let us see what you mean by nucleotides. What are the main components in the case of nucleotides? As we already seen that sugar, right? Sugar and base. Sugar and the nitrogen base is known as the nucleoside, right? So if the nucleoside is combined with the phosphate group by the ester bond, right? Ester bond, so it is known as a nucleotide. So we can define the nucleotide as the phosphoric acid esters, which means the ester bond formed between the phosphoric acid and the nucleoside is known as the nucleotide. Clear? So as you can see over here, this is our sugar, right? This is our pentose sugar, whether it may be ribose or deoxyribose sugar. So we are taking here, this is our sugar here. This will be our nitrogen base, any of the purine base or pyrimidine bases. So we can say that in the case of base, right? Base, there will be the nitrogen, right? There will be the nitrogen that is combining with, the first carbon of the sugar to form, an N-glycosidic linkage, right? N-glycosidic linkage, which is formed by the release of water molecule, right? Then, what is the another bond you are observing here? There is a phosphoester bond, right? So, this is our phosphoric group, that is double bond of OH. Again here, there will be the OH group. Again here, OH group. Then, you can see that there will be an OH group, right? In the case of sugar molecule, there will be a CH2OH group. Then, here will be the one of the hydrogen. So, one of the OH group present in the sugar combined with the another hydrogen present in the phosphoric acid to form water molecule. There will be a release of water molecule to form a bond known as phosphoester bond. Clear? Phosphoester bond. So when you are observing about the nucleotides, you can see there are two types of linkages are present. One is N-glycosidic linkage, which is formed between the nitrogenous base and the sugar, right? Then the next one, it is the phosphoester bond, which is formed between the phosphate group and the sugar molecule, right? Then the position of this particular group will be very important, right? The 5' position of sugar moiety, right? This is the fifth carbon having the OH group. So we can say that the fifth position OH group or the OH group in the fifth position are combined with the OH group of the phosphoric acid. Clear? Now let us see how these nucleic acids, right, either DNA or RNA are forming by the linkage between nucleotides, right? So, this is the structure of one part of the DNA or RNA. You can see that here, this is our sugar molecule, right? This is our sugar molecule. This is our sugar molecule. This is our phosphate group, and this is one of the nitrogenous bases, right? Nitrogenous bases. In general, there will be the glycosidic linkage, right? There will be the glycosidic linkage in between nitrogenous bases, right? In between nitrogenous base and the sugar molecule. So here you are observing that here, right, this is our CH2OH molecule. One of the OH present in the fifth position of sugar molecule, combined with the hydrogen atom present in the phosphoric acid, right? There will be the OH group, one of the hydrogen will combine with the CH2OH to form water molecule. By releasing the water molecule or the condensation process, it will form a phosphoester bond, right? Phosphoester bond. So in total, in the case of a nucleotide chain, you can see that there will be two ester bonds are present, right? There will be two ester bonds are present. So these ester bonds are termed as phosphodiester linkage. Phosphor stands for the phosphorus present in the linkage. Di means two ester bonds, Diester means there will be 2 ester bonds. Clear? Then the position of these particular group is very important here. 3 dash, right? 3 dash oxygen of 1 nucleotide. You can say this is 1 nucleotide, right? This is a 1 nucleotide containing phosphate group, sugar and the nitrogenous bases, right? So 3 dash position, right? This will be the 3 dash position of one of the nucleotide combines with the 5 dash position, 5 dash oxygen of the next nucleotide, 5 dash oxygen of the next nucleotide. Clear? Okay, I hope it is clear for everyone. In the case of nucleotides or in the case of a nucleic acid, either in RNA or DNA, nucleotides are combined together to form the polynucleotides or the nucleic acids chain. So, in the case of nucleotides, if they are combined together, we can say that there will be 2 linkages are present, right? There will be 2 linkages are present. One is the glycosidic linkage in between the nitrogenous base and the sugar, and another one is the phosphodiester linkage which is present in between the phosphate group and the sugar moiety, right? Clear? Now let us practice some questions based on the nucleic acid topic. Here is the first question. Which type of linkage is present in the nucleotide between base and sugar? Nucleotide between base and sugar. Option C, right? Okay, Kudaj Gari, you are right. Option 3, which means N-glycosidic linkage, right? So we have already seen that nitrogen, right? Nitrogen present in the nitrogenous base is combined with the sugar moiety, first position of carbon and then sugar to form the N-glycosidic linkage. So option 3 will be the correct answer. Then what about the next question's answer? Here is the next question. DNA and RNA contain 4 bases each. Which of the following bases is not present in RNA? Read the question carefully. Which of the following bases not present in RNA? Not present in RNA. Are you sure, Kudaj? Option B or C? Option 3, right? When you are discussing about the structure of RNA and DNA, you will be getting a detailed idea about the structure. So, if you are containing RNA, right, containing RNA, there will be 4 bases present. One is adenine, right? Then guanine. Third one is cytosine, right? Then fourth one, it is uracil in the case of RNA, right? Uracil in the case of RNA. But if you are considering DNA, instead of uracil, there will be the thymine base. Clear? So don't get confused in between the structure of RNA and DNA. This is the main difference between the RNA and DNA structure. The difference in the nitrogenous bases, right? So make sure that uracil will be there. You may keep in mind that uracil will be the nitrogenous bases which is present in the RNA, not in DNA. Clear? Yes, Deeptisha, you are also correct. Uracil. Okay, can we move to the next question? Again, here is the next question. The NH2 group is absent in. This question is based on the structure of nitrogenous bases. We have already seen the structures of 5 different bases, right? 5 different nitrogenous bases. We have to identify the nitrogenous bases which does not have the NH2 group. What will be the answer? Is there any guess? Now we will get the answer for the question. This is the structure of different nitrogenous bases. Which one of the following does not contain NH2 group? Here, right, in the case of cytosine, there is an NH2 group. So, here in the case of thymine, there is no NH2 group, right? There is no NH2 group. So, in case of our question, there will maybe answer like thymine or uracil. In the case of uracil also, there is no any amine group, right? There is no any NH2 group. Then in case of adenine, there is an NH2 group. In case of guanine also, there is an NH2 group, right? So, we may have the 2 nitrogenous bases which does not contain the amino group or the NH2 group, right? So, let us see, let's go with our question here. Okay, which one will be the answer now? Again, uracil is the right one, right? Uracil will be the correct one. In terms of nitrogenous bases, we can say that there are 2 nitrogenous bases, that is thymine and uracil, right? Thymine and uracil are the 2 nitrogenous bases that does not contain the NH2 group. Is that clear now? Okay, fine. Here is the next question. The sugar moiety which is present in DNA and RNA respectively are. The sugar moiety which is present in DNA and RNA molecules respectively. Which one will be the correct answer? Okay, we know that there will be the sugar which is present in the DNA, it is deoxyribose, and the case of RNA is ribose, right? So, Here you have to mention the anomeric position, right? Whether it is alpha or beta, along with their DNL configuration, right? DNL configuration. What will be the answer here? Four options are given here, right? You can guess any of the options. Just give me the guess answer also. In DNA it is deoxyribose, right? In DNA it is deoxyribose. So, option 2 will be the correct answer, right? Beta D2-deoxyribose. When you are discussing about the structure of, when you discuss about the structure of deoxyribose, right? Structure of deoxyribose and ribose, you can see that there will be the anomers having the beta position, right? Beta position. So, the sugar which is present in the DNA is beta D2-deoxyribose and the RNA it is beta D ribose. Is that clear? Here is the next question. The base which is present in DNA but not in RNA. Same repeated question, right? Read the question carefully, don't give me the false answers. Because this type of question we have already discussed. Which of the base which is present in DNA but not in RNA? Thymine, right? So we are discussing about the DNA. We have studied that thymine, which means option 4 will be the correct answer here. Thymine is present in the case of DNA, but in case of RNA, it will be the uracil instead of thymine. Clear? Here is the next question. The nucleosides are composed of, what are the main components which is present in the case of nucleosides? Not nucleotides, it is nucleosides. In question, it is asking about the nucleosides, not about the nucleotides. We have already seen the structure of nucleosides, right? It contains nitrogenous base and the pentose sugar, which means option 3 will be the correct answer. If you are adding any of the phosphate group, right? Phosphate group or phosphoric acid, you may get the nucleotide. Clear? Option 3 will be the correct answer. Here is the next question. The bases that are common in both RNA and DNA. The bases that are common in both RNA and DNA. While discussing this question, you may have the idea that there will be the repeating questions, right, about the nitrogenous bases present in RNA and DNA. So this is a very important topic in terms of nucleic acids. The bases that are common in both RNA and DNA. Adenine, guanine and cytosine, right? Adenine, guanine and cytosine, which means option 1 will be the correct answer. These are the nitrogenous bases which is common in RNA and DNA, right? RNA and DNA. Clear? Option 1 will be the correct answer here. Here is the next question which is based on the structure of nitrogenous bases. The nitrogen base that does not contain a keto or the oxo group in its structure. What will be the answer? Okay, these are the basic structures of the nitrogenous bases. Now you can identify which one is not having the keto group or the oxo group, which means C double bond group. Here, right, in the case of guanine, there is a C double bond group or the oxo group. Again, cytosine also there is a C double bond group or the oxo group. Then in case of thymine and uracil, you can see that there will be two C double bond group, right? There are the dioxo groups. The only one nitrogenous basis which does not having the oxo group or the keto group or C double bond group is known as the adenine, right? Adenine. So the structure of nitrogenous bases is very important. Which one is the answer here? Option 1, right? Yes, adenine will be the correct answer here. Here is the next question. In a nucleotide, the C1 of sugar is joined to, whether it is the N1 of a pyrimidine base or N9 of a pyrimidine base, N9 of both pyrimidine and purine bases, N1 of both pyrimidine and purine bases. We have already seen that there will be the N-glycosidic. Hi Pratibha. Okay, there is a glycosidic linkage is present between the base and sugar, right? Nitrogenous base and sugar. So, we have discussed about the two types of nitrogenous bases. One is purines and another one is pyrimidine. Whenever we are discussing about the types of nitrogenous bases, we have seen that there will be the glycosidic linkages. There will be the glycosidic linkages formed between the nitrogen of nitrogenous base to the sugar, right? So, if you are considering about the purine, there will be the particular nitrogen atom, right? Which is in the first or ninth position. If you are considering about the pyrimidine, there will be again particular position of the nitrogen atom that is combined to form the glycosidic linkage, right? What will be the answer here? Okay, what will be the answer here? For JEE Main session 2, right? Okay. Actually, I have no exact idea about the admit card. Maybe you can expect this admit card in the last week of the March, right? Which is the answer here? You are giving 4 different options. Which one is the correct answer? Option 1, right? So, if you are discussing about the pyrimidine base, you can say that in the case of pyrimidine base, there will be the first position, right? First position in the nitrogen atom. If you are considering about the purine base, there will be the 9th position of nitrogen atom combining with the sugar to form the N-glycosidic linkage. Is that clear now? Okay, fine. That's all about from my side for today. We have discussed about the details about the nucleic acid, right? We have seen that nucleic acid contain nucleotides, which is containing nitrogenous bases, sugar, and the phosphate group, right? Phosphate or phosphoric acid group. So there will be two different bonds are formed between the nucleotides. One is glycosidic linkage, that is N-glycosidic linkage. Then the next one is a phosphodiester linkage. So in terms of JEE Main examination point of view, you have a better idea about the structure of different nitrogenous bases. In the latest examination, in the January attempt also, you have already given the question about the structure of nitrogenous bases, about the structure of nitrogenous bases, right? Match the following. So again, there is a gentle reminder about the All India test series, which means this is the right time to take the mock test here, because the JEE Main second attempt is coming, right? Only few days are remaining for the second attempt. So you can take our AATS. You will get a 50% off by using the YouTube 50 off code. The link will be provided in the description. You can take that link and enroll just now. So in the next session, we will be discussing about the structure of DNA and RNA detailing. Till then, bye bye all of you. I hope it was very clear for everyone. If you really like the session, just hit the like button, subscribe our channel, and share this particular session to your friends also. Okay, we will be conducting the same class, right? We will be conducting the chemistry lectures in the same channel on every day. It is 5 to 6. So make sure that we will be attending the class daily, right? RBSc exams, right? You need to study well, right? There is no any other options. So many of you are asking about the session PDF. You have to join our Telegram channel, right? Infinity Learn JEE Telegram channel. You will get the session PDF there after every classes, right? Only 4 days are remaining, right? You didn't start it yet? Okay, try your level best. What you can do maximum, it will be your level best, right? So don't take too much stress or tension for the exams. So far we have discussed about the most of the important biomolecules in the biomolecules chapter. If anyone missed the classes, just go and revise the particular sessions, right? So I think we can wind up for today. If you have any doubt, you can ask now, otherwise we can wind up the session. Okay, what is your question? Pratibha? Okay, what will be the doubt? Just ask me fast. Because after this slide, there will be the maths lecture, right? Taken by Pratnya ma'am. Okay, I think we can wind up the session for today. Only 2 minutes are remaining. Okay, wait. In cellulose, glucose units are joined by glycosidic linkage between dash of one glucose unit and dash of the next glucose unit. Okay, we have already discussed about the cellulose structure in our first class, right? About the carbohydrates. Just go and revise the particular session, right? Just go and revise the particular session for the first class of the biomolecules. Is given in this particular channel, Infinity Learn JEE channel. If you still have any doubt after watching this class, just let me know in tomorrow, right? Clear? Is that clear now? Is it fine now, Pradeepa? Okay, fine. Because time, if you are going to discuss about the structure, no, it will be take little bit time to draw the structures also, and it's already time is going to over by 6 now. Just go and revise the session, right? After that, come on the next session. I mean tomorrow we can discuss. Tomorrow will be the last session of the biomolecules chapter. I will be covering the most important topics in the biomolecules also, along with the questions. Right? Clear? Okay, fine now. Anyway, thank you so much, all of you. Okay, thank you so much, all of you. Bye bye and take care.
No problem.